We'll move on to the next speaker, Richard Schlegel from Georgetown University. Give us a talk on clinical translation of HPV and cell culture research.
Well, I'd like to thank Dr. Sandberg and the organizers for the invitation to speak here, and I'm going to make a transition from quantum mechanics back to what Dr. DeMarchi was speaking about in terms of biology, and talk about what we've been doing with human papillomaviruses, as well as a new technology in cell culture called conditional cell reprogramming. And they do actually have an interface, and I'll explain the transition in that in a couple slides. As many of you know, the human papillomaviruses are involved in cervical cancer, and there's a vaccine for that right now. But some of you may not know that there's an increasing role for the papillomaviruses in many types of cancers, not only cervical, but vaginal and penile cancers, and as well known from Michael Douglas's case, oral, tongue, and throat cancers, and the unfortunate case with Farrah Fawcett, who died from anal cancers, and some increasing evidence for its role in skin cancers. So obviously it's an important etiologic agent in about 5% of human cancers, a very important target for considering therapies or preventions. This represents the human papillomavirus, and I'm not going to go into detail on it, but it's really basically a protein shell consisting of two proteins, L1 and L2, but the predominant protein is the L1 protein, and that forms this core over here. Let's see if I have, here we go, this core here. Inside that is a double-stranded DNA molecule. It's circularized. And the virus in general is a very simple one. If you take that DNA, and I'll show it structurally on the next slide, represented over here, this is the genome, the double-stranded DNA, and it's got early genes that have role in transformation and causing the problems with cancer. And these are the two proteins, the capsid proteins, the structural proteins, and L1. This is the one that we did early work in about 1993. It only took about 15 years to get vaccines after that discovery. Being able to make that protein in cellular systems where it could be used successfully as a vaccine. And essentially, if you overexpress that protein, the L1 protein, either in yeast or in baculovirus systems, insect cells, what happens is it spontaneously folds and makes these structures here that are called virus-like particles. So this is the L1 protein. First it forms pentamers, and then it forms these capsid structures, which are 72 pentamers. And it very closely, as you can see, mimics what a virus particle looks like, except this has DNA in it. That's why this looks full, and that's why these are empty. But this is the vaccine that's available right now from Merck and GlaxoSmithKline. And that is completely non-infectious and safe for human populations and very efficient, as I'll show in a couple slides. So how it works is when you inject those particles, you make antibodies against that L1 protein, which exactly mimics what the virus looks like. Those antibodies bind to the surface of the virus here, and they completely protect against infection of cells. They block the attachment, and even if the virus gets inside, it can't infect the cells. And I won't go through the biology of this, but unless these viruses can get access to what are more like stem cells down here, you can't get infection. And so it's very important that we make antibodies in our saliva, in vaginal secretions, and those antibodies are the ones that induce the IgG that block infection. As I said, there are two companies out there currently marketing the vaccine. There were 4 groups involved in different aspects of this vaccine. Georgetown's group, I was working with Bennett Jensen and Sinjay Gim. Queensland, Ian Fraser was working, Bob Rose's group at Rochester, and Doug Lowy and John Schiller at the NIH. And via complicated patent issues and 10 years of negotiations between companies and everyone else, finally these things got marketed, and there's a sharing of profits and that come from these 2 vaccines. The important thing I'd like to focus on here is both of these vaccines are very effective. They're 100% effective in preventing persistent infection, which is a precursor to cancer. And there's a new one coming out from Merck, Gardasil 9, that will be predicted to prevent about 90 to 95% of human cancers. And that would be at all anatomic sites, not just the cervix. So the vaccine is very effective, as I showed on the previous slide, and this is the documentation. If you study a population of women that have been vaccinated, 30% of them vaccinated, it prevents 50% of the transmission of the virus. So there's herd immunity going on in this particular case, but shows you how clinically efficacious the vaccine is. And finally, the new generation one coming out, the Gardasil 9, for example, if you look at the worldwide impact, we're going to, we'll go back here a little bit. We'll be able to prevent 250,000 deaths per year just from cervical cancer. That's not including the increasing problems we're having with oral cancers. As a consequence of our studies that we were doing with HPV, we were studying the early genes on that diagram I showed that are involved in transformation. And basically what the virus does is it immortalizes cells. It makes them grow forever, which is the first critical step in cancer. And as a consequence of our studying immortalization, we came upon a way to, quote, conditionally immortalize human cells. So we can now grow cells from any epithelial surface from us or other animals indefinitely in vitro, and it opens up a wide number of studies that we can do. So for example, we can even use frozen tissue and fresh tissue that we get from the pathology department, my department, that we get from the surgical suite, needle biopsies. It's working very well for that. And the real breakthrough here is using two components, special feeder cells that make factors that allow the epithelial cells to grow. And secondly, I won't go into this, a chemical inhibitor of a specific pathway in the cell, a Rho kinase pathway. Those two factors together allow cells, this shows prostate, breast, tracheal cells, lung cells, and liver cells that can grow indefinitely as long as you maintain these conditions. The interesting thing in this case, this happens to be a colony of breast cells, I believe, in this particular case, or prostate it could be. But here's prostate cells under those conditions. This is time, they grow forever. And these are mammary cells that grow forever. This also, I won't go into the technology, but it works on colon, it works on many different tissues, including neuroendocrine. So we're being very successful growing neuroendocrine cells, including those from the islets of the pancreas. And so what we're really focusing on right now is understanding this process, but also in utilizing it for regenerative medicine and precision medicine. So, for example, if you take those conditions away from a cell now, the ROCK inhibitor and the feeders, here they are represented up there. Here are cells growing under our conditions from both skin and from the trachea. If you take those conditions away and put them on an air-liquid interface, the skin cells form skin, beautiful skin, and the tracheal cells form lung tissue with ciliated cells. And I'll show a couple in vivo pictures of this, which is even more dramatic in my mind. For example, if you take normal conditions for growing skin cells and you transplant them onto a mouse, they form a skin structure that looks like this, which looks great, but what you're lacking here is hair follicles, sebaceous glands, sweat glands that really anchor the epidermis to the dermis. And so this is a relatively fragile transplant. With the conditions that we're using, we're developing those structures which allow the anchoring of that epithelium to the underlying structures. So there's great potential for skin transplantation using this. With regard to the lung, we see similar maintenance of the biology. These represent tracheal biopsies from a patient with cystic fibrosis. And what you see over here, the white peaks over here represent cilia, and the green spots over here represent cells making mucus. So the technique we have actually maintains many different types of stem cells, and we certainly have at least two here, the mucus-forming cells as well as the ciliated cells. Probably even most important, if you put these into a different environment now and the tracheal cells, rather than being flat, form this structure that looks very much like the lung. This would be airway in here. What's in here right now is mucus made by the mucus cells, and these are the ciliated cells, and you can't see them very well right here, but they're all along here. But if you blow that up and you look at them, the cilia are functional, and so they beat in a coordinated way. They all have a slow and fast stroke so that the mucus is swirling around. These are the cells that are clearing the mucus in our lungs and preventing infection by organisms, viruses and bacteria that we entrap. So we can not only use this method for regenerative medicine, but also for precision medicine. And this is the example I'd like to show here, where we have a biopsy of a patient who had a lung cancer, actually induced by human papillomavirus. And what you can see here is that within 36 hours, we had normal cells from that patient growing up, as well as tumor cells from that patient. And this is very important. The technology works on tumors as well as normal cells. And so that allows us then to do studies looking for drugs or therapeutic approaches that allow us to show it doesn't kill the normal cell, but does kill the disease cell, or at least in some cases for regenerative medicine, make the disease cell behave like the normal cell. And so we've grown these cells up, and within 8 days, we were able to do some drug screening studies. And here what you see is we've used the normal and tumor cells screened against one drug that was presumed to be effective, and the patient was going to be transferred to NIH for that therapy. Another drug that works on HPV cervical cancers, and then one here, verinostat, which is an HDAC inhibitor. I won't go into that, where there is a difference between the response of tumor cells being sensitive and the normal cells less sensitive. And based upon this experimental approach, the clinicians, since this is an approved drug for treating other types of cancers, the patient was treated and had a 30% regression within 3 months and was stable for almost 3 years. And considering that this is a rapidly progressive disease, this is a major step forward in our being able to use this technology for functionally screening what the tumor cells are responsive to. Not just, it'll be probably combined with the genetic approaches, which Dr. DeMarchi was mentioning. We certainly need to know what genes are altered, but we also need to know functionally how those different pathways interact and alter the biology of the cell. So this technique is being used at many groups. We've trained about 24 laboratories at Georgetown. Jeff Engelman's group at Harvard is using it, and we've been interacting with him. And he's shown very effectively that this technique can be used to screen and identify drugs that are effective against cancers, even in those cases where the genetics don't identify a target. And that's the critical thing. Where the genetics identify targets, the technique is helpful in essentially informing it and validating it. But in this particular case, you can also identify drugs that work even though the genetics don't give you any hint. And so what I'd like to end on is really just showing you how this technology is being transferred, like our HPV technology. A new startup company was recently formed last April. The CEO is Brian Pollock. He used to be the president of the American Type Culture Collection. This company is really a platform company. What they're doing is focusing on regenerative and predictive medicine. And so they will probably be interacting or having spin-offs from their company, because obviously this is a very wide platform, requires a lot of expertise. And even if you think about regenerative medicine, each tissue has its own specific requirements for addressing clinical issues. And this company, if I go back here, whoops, I'll take one step back. Propagenics is already beginning to work with another company that's forming up in Boston called Leap Oncology. And their main goal is, and their belief, is that by 2020, in 5 years, functional cell-based testing is really going to be the guiding therapy, and it's going to become really the standard practice in medicine in the future. So anyway, I guess the tie-in between the HPV work and as well as our work in cell technology is not only they're related biologically, but they're both leading us in directions that lead to commercial development. And I think I will stop there. Right. Well, actually, that dogma is still true, is when we have these highly proliferative states, the cells are in an undifferentiated stem-like state. There are a lot of markers that are turned on that resemble adult stem cells. Then when you take those conditions away, which is a major advantage when you think about induced pluripotent stem cells, you have to work very hard to get stem cells to go in the direction you want. In our case, when we take those factors away, there's a memory, and those cells go back to form the tissue from which they were derived.